Hi, I'm Chris, and in this video we're going to talk a little bit about using Punnett squares to understand inheritance in terms of blood type. So when you're dealing with blood type, we have type A, type B, type AB, and type O. Now you may have heard of someone who might be like type A positive or A negative, and that's incorporating something else that's called Rh factor, which we'll talk about in our next video. But for this one, we're just getting an introductory look at these four blood types and what that actually means. So hopefully you've already been introduced to Punnett squares and the way that different kinds of alleles combine together to give you different genotypes, which is, think of that as like the letters, whether it's big A, little a, or big A, big A. And then correspondingly, that results in a particular phenotype, whether you're looking at, something like brown hair versus blonde hair, or in some of the examples you might have seen in class, like a red-eyed or white-eyed Drosophila, which is a fruit fly. So those are the kinds of examples we're talking about. We're going to see how that applies with blood type. So with blood type, you have these different alleles. You have A, B, and O. And both A and B are going to be dominant over O. You can think of O almost as like a blank. So instead of thinking of it as AO, think of it as almost A blank. And that's going to help you remember that either A or B, when paired with that blank, when paired with O, is going to be dominant, is going to determine what the blood type outcome will be. Let's take a look at an example of how this works in a few different cases. So if you look at these four Punnett squares, you might notice that this is someone who's type A being crossed with someone who's type B. Type A, type B. All four of these, in fact, are type A and type B, type A crossed with type B. The difference in these, though, is that here we have a homozygous dominant, big A, and same thing for B. Here we have AO and BB. Here we have AO and BO, and here we have AA and BO. So we're going to see how these different changes in alleles actually change the blood types of the offspring from these different crosses. So let's take a look. In this first example, for each one, we're going to have an A and a B, which means in that situation, we have type A and type B being crossed, and all four offspring, in this case, no matter what, there's a 100% chance that the offspring get type AB. Let's see how that changes when we incorporate an O as one of the alleles. So here we have AB, but here we have BO. Here we have AB, here we have BO. So in this case, you had 100% AB. Let me just write that in. Here we had 50% AB, and 50% of the offspring in this cross would be type B. So 50% AB and 50% type B for that one. Now let's take a look at what happens here. So AB, AO, BO, and OO. So here you actually have all four blood types possible in the offspring. So you have 25% chance of being type AB, 25% chance of being type A, 25% chance of being type B, and 25% chance of being type O. And this is pretty much what we think of when we think of two heterozygotes in a cross, is that what happens in terms of the combinations of alleles that you get, you have 25% chance of each type of outcome as we see it here. And then this last example, we have AB, AO, AB, and AO. So this is similar to that second example. 50% will be type AB and 50% will be type A. So with blood type, just remember that to be type A, you don't necessarily have to have two copies of that allele. You can be AO, and the phenotype will still be blood type A. And same thing for B. There's also something else shown here, and I don't know if it's a vocab term that you've seen yet, but as you get introduced to it, think of this example. There's something called incomplete dominance, which basically means that more than one allele can code for a particular phenotype that gets expressed at the same time. So if you're A and B, it's not that one is dominant over the other, but both of them together form a separate phenotype, which is type AB. And that's what we see in any of these examples where you're looking at type AB. A is not dominant to B and B is not dominant to A, but together they form a separate phenotype, a separate blood type, type AB. For more, please feel free to watch our other bio videos, and thanks for watching.